Good afternoon, everyone. We are here for the regulatory affairs session with the topic WHO guidelines on observational studies in traditional medicine research. In the previous session, we discussed regulatory framework of African agencies SAFRA and CAPA and its harmonization with case studies. In today's session, Rutuja Shinde ma'am will deliver keynotes on WHO guidelines on observational studies in traditional medicine research. Over to you, ma'am.
Thank you, Neha. Good afternoon, everyone, and welcome. We begin by exploring what traditional medicine means in today's healthcare context. Traditional medicine encompasses a vast range of health practices, knowledge, and beliefs that rely on natural resources, plants, animals, minerals, along with spiritual and manual therapies. So, this system evolved over centuries within distinct cultural frameworks, shaping healing approaches around the world. So today we'll begin with the WHO guidelines on observational studies in traditional medicine research. So understanding traditional medicine in modern healthcare. So WHO Health Organization recognizes traditional medicine as an integral component of healthcare for millions, especially in low and middle income countries. As a modern health system expand, understanding how these traditional approaches coexist and complement conventional medicines has become increasingly important. The growing need for evidence-based research. So here we highlight why rigorous evidence is crucial. So roughly 80% of people in developing nations rely primarily on traditional medicine. 170 WHO member states report active use of traditional and complementary medicine, contributing to a market valued at 83 billion US dollars, projected to reach 115 billion by 2027. So these figures reveal not only a widespread reliance but also major economic and public health stakes. So to ensure safety, effectiveness, and policy relevance, research on traditional medicine must meet the same methodological and ethical standards applied to other medical sciences. Then, why observational studies matter? So, observational studies are vital because they examine a real world practice without intervention. Unlike randomized control trials that impose strict protocols, observational designs respect how traditional medicine is naturally delivered within communities. They capture authentic data on safety outcomes and uses pattern. Through these studies, we can translate everyday practice into reliable evidence while preserving cultural integrity. So this breach between lived tradition and scientific evaluation is the heart of the WHO guidelines. Then core principle of WHO guidelines. So WHO set out four guiding principles for observational research in traditional medicine. First is scientific rigor, employing robust methodological standards based accommodating traditional medicine paradigms. Then ethical integrity that protect participants, communities, and traditional knowledge. Then cultural sensitivity, respect the indigenous practices and belief systems. Then practical applicability, generate finding that can directly inform health policy and service delivery. So, these principles form the ethical and scientific backbone of all subsequent recommendations. Then, types of observational studies, understanding the methodological landscape for traditional medicine research, cohort studies in traditional medicine. So, observational research takes several forms, each suited to different questions. We will briefly outline the main design: cohort study, case control study, cross sectional survey, and ecological studies. So, understanding their distinctions help us choose the correct framework for a specific traditional medicine question, whether exploring long term effects, identifying risk factors, or describing patterns of use. So, first one is cohort study in traditional medicine. So, cohort study follows groups of individual over time to examine how exposure, such as herbal formula or acupuncture regimen, affect outcomes. So, design can be prospective or retrospective, but both involve comparing exposed and unexposed participants. Because they establish a temporal relationship, cohort studies are strong for linking cause and effect with a real world practice. However, they require a sustained follow up and careful tracking. No small task in community setting. So, key characteristics are prospective or retrospective design, long term follow up periods, multiple outcome measurements, then comparison between exposed and unexposed groups. So, this studies excel as establishing the temporal relationship between traditional medicine use and health outcomes. Then case control studies, investigating associations. So case control design start with the people who already have an outcome of interest, say improved respiratory health, and compare them with the controls who do not. Researchers then look backward to evaluate exposure to traditional treatments. By calculating odd ratios, we estimate the strength of relationship between the practice and the outcomes. So, identify cases, select individuals with a specific health outcomes or condition of interest that match control, choose comparable individuals without the outcome match on key characteristics. Then, access exposure retrospectively, examine traditional medicine use patterns in both groups, and then analyze the association, calculate odd ratios to determine relationship between exposure and outcome. So they are efficient of rare conditions and can quickly generate valuable insights, though they rely heavily on accurate recall and matching. Then cross sectional studies, snapshot research. So, cross sectional study takes a single snapshot in time, measuring both exposure and outcome simultaneously. So, it tells us what is happening now. For example, how many people currently use a certain herbal remedy and report relief. So, this study are cost effective and useful for estimating prevalence or generating hypotheses, though they cannot determine causations. So this serve as a foundation for planning more in depth research. Then ecological studies, population level insights. So, ecological studies shift focus from individuals to population. So, they compare rates of traditional medicine use and health outcomes across regions or time periods. Such analysis reveal broad trends. For example, reduce infant mortality in areas with integrated traditional midwifery. However, we must beware of the ecological fallacy. Relationships seen at the population level may not hold true for individuals. So, used wisely, these studies guide policy level thinking. Then planning your study. So planning is the cornerstone of sound research, essential steps for designing rigorous observational research. So before data collection begins, clarify your objectives, methods, and ethical pathways. A well-designed plan ensures the study answers real questions and meets scientific standards. So this slide introduced the five key steps that follow. First is identify knowledge gaps, then formulate specific questions, establish primary outcomes, then determine secondary objectives, and consider practical implication. So start by reviewing existing literature to identify knowledge gaps in traditional medicine practice. Formulate specific answerable research questions and define primary and secondary outcomes. Always consider how your results will be applied. Will they inform policy, guide clinicians, or support community practice? So, clarity at this stage prevents wasted effort later on. Then, selecting appropriate study population. So, defining who to include and exclude is critical. So, inclusion criteria specify participants who fit your research focus by age, condition, or treatment use. So, age ranges appropriate to the research questions, specific health conditions, or wellness status, traditional medicine exposure or use patterns, then geographic location and accessibility, and lastly ability to provide informed consent. Then exclusion criteria protect validity by removing factors that could distort results, such as coexisting diseases or language barriers. So balance representativeness with feasibility and always ensure informed consent if possible. Then sample size considerations. So adequate sample size gives a statistical power to detect the real effects. Work with a biostatistician to estimate numbers based on expected effect sizes, variability, and dropout rates. Because traditional medicine treatments can be highly individualized factor that heterogeneity into your calculations. So underpowered studies waste resources. Oversized ones can strain budgets and communities. Then data collection method. So reliable data collection builds credibility. Combine method as appropriate, structured interview to capture experience, standardize questionnaires for quantitative data, medical records for objectivity, and clinical examinations for direct measurements. So, blending subjective and objective sources mirrors the holistic nature of traditional medicine practice. Then ensuring data quality. So data quality relies on training standardization, quality checks, and validation. So, train collectors in both protocols and cultural context. Then use standard operating procedures to ensure consistency and validate the data through cross checking. Then frequent quality reviews, catch errors early, and maintain integrity throughout the study, and validation cross check data through multiple sources and methods to ensure accuracy and reliability. Then ethical considerations, protecting participants and respecting traditional knowledge. So, ethics in traditional medicine research extend beyond individual consent. We must also respect a cultural ownership of knowledge and protect community interest. So the following slides outline key ethical areas: inform consent, vulnerable populations, traditional knowledge, and confidentiality. So, informed consent, a fundamental requirement, ensures a voluntary participation. So, participants must understand the study's purpose, procedure, risk, and benefits in their own language. Explain that withdrawal is allowed without penalty and confidentiality is assured. Adapt consent methods to local culture, for example, through community elders or oral processes when literacy is low. Then protecting vulnerable population. So extra care is needed for children, indigenous peoples, and the economically disadvantaged. So seek parental consent and child assent when appropriate. Engage community leaders for collective approval and avoid correction through financial incentives. Then, ethical balance means enabling participation while safeguarding dignity and autonomy. Then protecting traditional knowledge. So traditional knowledge belongs to communities, not individuals. Researchers must respect intellectual property and benefit sharing before publication. Secure permissions and clearly define ownership of data and findings. Always acknowledge knowledge holders and maintain transparency on how their wisdom is used. Then ethical review board approval. So every study requires a formal ethical clearance. Prepare a comprehensive protocol, submit it to the relevant review board, respond to queries, and obtain a written approval before field work begins. Continue reporting progress and amendments throughout the study. So, this accountability builds trust between researchers, participants, and institutions. Then confidentiality and data protection. So protecting participant privacy is a non-negotiable aspect of ethical research. Store data in secure password protected systems and use encrypted channels for sensitive communication. Restrict access to authorized personnel only, and de-identify data sets before analysis. Develop a clear retention and destruction policies. So always balance openness in research with strong privacy safeguards. So this protects both participants and your study credibility. Then measuring traditional medicine exposure, documenting practices accurately and comprehensively. Accurate exposure measurement is vital. Uh, Traditional medicine practices are rarely standardized, so documentation must be detailed. Specify the modality, herbal therapy, acupuncture, manual, or spiritual practice, and capture practitioner background, treatment duration, and frequency. So, such precision ensures that latter readers or replications truly understand what was studied. Characterizing traditional medicine interventions. So, traditional medicine often involves complex, individualized interventions that differ substantially from standardized pharmaceutical treatments. Comprehensive documentation is essential for meaningful research. First is modality types, so specify whether intervention involves herbal medicine, acupuncture, manual therapy, spiritual practices, or combinations. Then practitioner details, document training experience, credentials, and traditional lineage of providers. Then treatment specifics, record ingredients, formulation, technique, duration, frequency, and dosage where applicable. Then individualization, note how treatments are adapted to individual patients' condition and constitutions. Then herbal medicine documentation. So where herbs are involved, identify every botanical scientifically. Record the Latin name, family, plant part used, preparation, process, dosage, and route of administration. Maintain voucher specimens in herbariums and quality control certificates. So this records make a result reproducible and safeguard against misidentification or contamination. Then capturing a treatment pattern. So traditional medicine often involves a variable treatment patterns that evolve based on patient response. Documenting these dynamic approaches requires sophisticated data collection strategies. So unlike fixed drug regimens, traditional treatments evolve with a patient response. So key dimensions are initial treatment selection, rational modifications during treatment course, concurrent use of multiple modalities, treatment discontinuation or completion, adherence patterns and barriers, and co-use of conventional medicine. So capturing this dynamism paints an authentic picture of real world traditional care. Then accessing treatment fidelity. So fidelity ensures that outcomes truly reflect the intended treatment. Monitor patient adherence, practitioner consistency with the protocol, and follow-up completion. Set quantitative benchmarks. For example, minimum 80% adherence or retention. Regular fidelity checks preserve internal validity. Then outcome assessment, measuring health effects and treatment success. So selecting appropriate outcome measures. So outcome selection should honor both biomedical and holistic traditions. Measure not only clinical indicators, lab values or symptoms, but also patient reported outcomes such as well-being and functional capacity. Then include safety outcomes and healthcare utilization, use of health services, hospitalization, and economic impacts of treatment. So, balanced matrix provides a complete view of impact. Then validated measurement instruments. So use validated tools to ensure reliability and comparability. So instruments like SF36, EQ5D, or WHO QOL access health and quality of life across cultures. Then validate measures, reduce bias, and increase confidence in findings. So avoid ad hoc questionnaires unless rigorously tested. Then timing of outcome assessment. So collect outcomes at 4 points: baseline, before treatment initiation to establish starting point. Then during treatment, regular intervals to capture response patterns. Then treatment completion, immediate post treatment assessment. Then follow up long term outcomes at predetermined intervals. So strategic timing captures both immediate effect and sustained benefits or harms of traditional medicine intervention. Then accessing adverse events. So systematic safety monitoring protects participant and enriches data quality. So use structured interviews to capture events, grade severity, evaluate casualty, and document management. Active surveillance regularly ask participants about potential adverse effects using structured queries. Then severity classification, grade events using standardized scales like mild, moderate, severe, or life threatening. Then casualty assessment, evaluate likelihood that observed event relate to traditional medicine exposure. Then documentation record comprehensive details including onset, duration, management, and outcome. And reporting, report serious events to ethics committees and regulatory authorities promptly. So this transparency strengthen trust in traditional medicine research. Managing confounding, addressing factors that obscure a true relationship. So confounding can blur true exposure outcome relationship. So understanding confounding in observational studies. So because randomization is absent, identify potential confounders early: age, gender, then socioeconomic status, co-treatments, disease severity, and lifestyle factors. So design strategies and analytical adjustments will control their influence. Then common confounders in traditional medicine research. So list typical confounders explicitly: demographics, economic level, concurrent medical care, and baseline health behaviors. So age influences both treatment choices and health outcomes. Then gender affects disease prevalence and treatment seeking. Then lifestyle factors, diet, exercise, smoking affect both exposure and outcomes. Then socioeconomic status determines healthcare access and health behaviors. Then disease severity, baseline health status influences treatment selection, and concurrent treatments, co-use of conventional medicine affects outcomes. So this proactive approach limits distortion of findings later. Then design strategies to control confounding. So two classic strategies: restriction and matching. So restriction narrows the participant pool to similar characteristics, matching pairs, exposed and unexposed participants with the same key variable. Um, so advantages are simple to implement, eliminates confounding by restricted variables. Disadvantage are reduced generalizability, may limit sample size. Then matching, for each exposed participant, select one or more unexposed participant with identical values for confounding variables. The advantages are balances confounders between groups, increase statistical efficiency. The disadvantage are cannot examine match variables as risk factors. May be difficult to find suitable matches. So each method simplifies interpretation, but can reduce the sample size or analytical flexibility. So choose carefully. Then analytical approach to confounding. So after data collection, use a statistical control methods that are stratification, uh, multi-variable regression, propensity score methods, and sensitivity analysis. So stratification analyze data separately within a strata defined by confounder levels, then combine results appropriately. Then multivariable regression include confounders as covariates in regression models to adjust for their effects simultaneously. Then propensity score method calculate probability of exposure given confounders. Then use scores for matching stratification or weighting. Then sensitivity analysis assess how robust findings are to unmeasured confounding through various analytical scenarios. So these tools allow clearer interpretation of traditional medicine effect. Then addressing indication bias. So indication bias arises when seeker patients are more likely to seek traditional care, making outcomes appear worse. Control this by measuring baseline severity restricting to comparable illness levels or using advanced models such as instrumental variables. So recognizing this bias is a key to fair evaluation. Then statistical analysis, extracting meaningful insights from observational data. So begin every analysis with a descriptive summaries, demographics, exposures, outcomes, missing data patterns. So then move to inferential test, chosen according to data type and research questions. So clear systematic analysis plans prevent data-driven bias and enhance transparency. So descriptive analysis, understanding your data. So describe who your participants are, how they use traditional medicine, and what outcomes were observed. So, key descriptive statistics are participant demographics and characteristics, traditional medicine exposure, prevalence, and patterns, outcome frequencies and distributions, covariant summaries, missing data patterns, follow up time distributions. So this contextual overview support interpretation and detects um, anomalies early. So descriptive statistics are not just preliminary; they ground the entire study narrative. Then choosing appropriate statistical test. So use a test that match outcome types. T-test or ANOVA for continuous variables, chi-square or logistic regression for binary outcomes, Cox regression for time to event data, and mixed models for repeated measures. So report effect estimates, confidence intervals, and ensure assumptions are made before interpretation. Then regression modeling strategies. So build models step by step. Start with a univariable analysis. Include theoretically relevant confounders. Check multi collinearity and validate model fit. First is model building. So begin with unvariable analysis, then develop multivariable models, including confounders and clinically relevant covariates. Consider interaction when theoretically justified. Then variable selection, base inclusion on a priori knowledge and theoretical considerations rather than purely statistical criteria. Avoid over fitting by limiting variables relative to sample size. Then model diagnostics, access assumptions through residual plots, goodness of fit test, and influence diagnostics. Check for multi collinearity among predictors and interpretation, present effect estimates with confidence intervals. Interpret in a context of study limitations and clinical significance, not just statistical significance. So, transparent modeling process is essential for credibility. Then, handling missing data. So, missing data can bias result. Classify the mechanism, that is MCAR, MAR, or MNAR, that is missing completely at random. Missingness unrelated to any variables least problematic but rare. Then missing at random, missingness related to observed variables but not the missing values themselves. Then missing not at random, missingness related to unobserved values, most problematic for inference. Then analytical approaches, complete case analysis, analyze only participants with a complete data, valid if MCAR but loses power. Then multiple imputation, create multiple data sets with a plausible imputed values, analyze each pool results, preferred methods for MAR. Then sensitivity analysis, examine how results change under different missing data assumption. So document assumptions and perform sensitivity checks to confirm robustness. Then survival analysis for time to event outcomes. So for outcomes such as recovery time or mortality, survival analysis handles censoring effectively. So use Kaplan-Meier curves to visualize long rank test to compare groups, and Cox proportional hazards models to adjust for confounders. Always verify proportional hazard assumptions before drawing conclusions. Then subgroup and interaction analysis. So subgroup analysis explore whether effects differ across categories such as age, gender, or disease severity. Pre-specify these subgroups in your protocol to avoid data dredging. Then when testing interactions, use formal statistical terms like p-values, comparing p-values across subgroup. Um, Use statistical terms in regression models rather than comparing p-value informally. Then interpret cautiously. Subgroup findings are exploratory, not definitive. Then interpreting and reporting results, communicating findings accurately and transparently. So interpret the results with precision and transparency. Present both effect size and 95% confidence intervals to show magnitude and uncertainty. Avoid over reliance on p values. So discuss what the data truly indicate about real world effectiveness and safety of traditional medicine intervention. So clear reporting builds scientific trust. So effect measures in observational studies, different effect measures quantify associations between traditional medicine exposure and outcomes. So choose measure appropriate to your study design and outcome type. So, risk ratio for cohorts, or ratio for case control, or rate ratio for time based outcomes. So, translate these numbers into clinically meaningful statement. Risk difference are often the easiest for policymakers to interpret. Then confidence interval and p-values. So confidence intervals express precision. Wider intervals mean more uncertainty. Remember, statistical significance does not equal clinical importance. Report both significance and practical impact. So encourage readers to focus on the real world meaning of your finding. Then accessing causality in observational research. So observational studies demonstrate association, not proof of causation. So observational studies demonstrate association but cannot definitively prove causation. Apply established criteria to access evidence for causal relationship. Temporality does exposure precede outcome, essential criterion for causation. Strength of associations, stronger associations are more likely causal through weak associations can also be causal. Dose response, dose increasing exposure produce greater effects, supports causality. Then consistency are findings replicated across different populations, settings, and study designs. Biological plausibility, is there a credible mechanism linking exposure to outcome? Specificity, is exposure associated with the specific outcomes rather than the multiple diverse outcomes? So, evaluating each helps determine whether your association might indeed be causal. Then transparent reporting standards. So follow established reporting frameworks to enhance reproducibility. Use the STROB statement, that is strengthening the reporting of observational studies in epidemiology. Comprehensive checklist for cohort case control and cross-sectional studies. Then record statement, reporting of studies conducted using observational routinely collected health data extends STROP for studies using existing databases. Then STROM ID, that is standards for reporting of observational studies in medicine indigenous data. Additional considerations for research with indigenous people. So, adhering to this ensures your work meets international expectations. Then, acknowledging limitations. So, no study is perfect. Discuss limitations openly, whether design related, measurement based, or analytical. Acknowledging this shows integrity and helps others interpret findings appropriately. So, transparency about weakness increase credibility rather than diminishing it. From evidence to action. Have to discuss this point. So, design limitations, observational research cannot establish causation and may suffer from residual confounding. Clarify these limitations in your discussion. Explain what steps you took to minimize bias and what uncertainties remain. Then, measurement issues. Measuring traditional practice accurately is challenging. Address possible recall bias, observer bias, or inconsistent exposure recording. So, discuss how this might affect your conclusions and how future studies could improve them. Then generalizability reflect on whether your findings apply beyond the study setting. Cultural diversity, treatment variability, and practitioner skill can influence external validity. Clarify to whom your results are most relevant. Then analytical decisions shape conclusions. So explain why certain variables or models were chosen, and how sensitivity analysis confirm stability. So this openness lets others trust and replicate your method. Then from evidence to action. So move from results to implications. Highlight how findings can inform clinical practice, public policy, and integration of traditional and modern systems. Differentiate proven effects from areas needing further research. Evidence is meaningful only when translated into real world improvement. Then building evidence through replication. So single study cannot stand alone. Replication across diverse settings validates or refines findings and uncovers contextual nuances, encourage data sharing, prospective registration, and publication of null results. So these all strengthen the global evidence base. Then facilitating replication, provide a detailed protocols, make a identified data accessible when ethical, and report negative findings transparently. Such openness accelerates scientific progress and builds collective confidence in traditional medicine research outcomes. In future directions in traditional medicine research, so the future is promising. Emerging areas include machine learning for complex data, integration science for blending traditional and modern systems, then mechanism research or mechanistic studies to understand biological pathway, and comparative effectiveness research. Then implementation studies will then translate proven practices into diverse healthcare settings. Then advancing global health through evidence. So uh, in closing, rigorous observational research in traditional medicine protects patients and informs policies. So, by combining scientific rigor, ethical integrity, and cultural respect, we contribute to equitable global health. Traditional medicines, heritage, and modern research standards need not conflict. Together, they enrich healthcare for all. So, the journey towards comprehensive traditional medicine evidence continues. Your research contributes to this vital global effort. So, thank you. Over to you, Neha.
Thank you, ma'am. There are some questions. First question is, what is the difference between missing completely at random and missing not at random? And why does this distinction matter for data analysis?
Missing completely, random, and
Missing not at random.
so by it is not I think it is not necessarily when designed rigorously and transparently. They provide complementary evidence, especially on safety and utilization. So, multi variable regression, propensity score techniques, and sensitivity analysis are commonly applied. So, this I think, credible measure across the population, improving comparability and scientific acceptance.
Another question is how can researchers protect traditional knowledge from exploitations?
So, traditional traditional knowledge is, through standardized operating procedures, through training and regular audits. So, by I think this way they can protect, and also through co-authorship, acknowledgement, data access, and equitable sharings of any commercial or policy benefits. So this is how they can, traditional knowledge holders benefit from research outcomes.
Okay, thank you, ma'am.